Hi everyone, welcome back. So gel electrophoresis is a really useful way to look at and analyze DNA. But in order to do gel electrophoresis, we have to have DNA to analyze. And a lot of that research starts with identifying a specific region of DNA and amplifying it many times, creating many millions of copies. And then those copies become the things that we analyze through gel electrophoresis. Now, this tactic is really useful because it gives us many copies of a specific region of DNA to do many different things. We could identify a specific DNA sequence, we could sequence it to figure out the A's, T's, C's, and G's that make it up, and therefore also figure out what it codes for. We could analyze how that gene actually works and look at its architecture. Does it have exons? Does it have introns? Different things like that. We could look and see how it relates to other genes, both in a functional context, as in what does this gene do compared to other genes, and also in a relatedness context, in an evolutionary context. How related is this gene to other genes or this gene in other organisms? We can even use this to identify what organism the gene came from. For example, we could take a sample that we did not know what the organism was, and say that it was a pathogen that came from a sick patient. We could use a specific gene that came from that organism to identify it to species. Now today, to do that, we use a process called polymerase chain reaction, or PCR. This is a DNA amplification method. So this is going to create many millions and millions of copies of a single gene, a specific region of DNA, starting from just a few copies. Now, PCR should be fairly simple for us to understand because we have already explored the concept and process of DNA synthesis inside a cell. Now, the process is going to be very similar, but the main difference is that it's performed under laboratory conditions in a test tube. It's not going to be made inside of a cell. So we're going to have special enzymes that we're going to use to achieve this. And we're going to do this reaction of doubling the DNA that we have many times, 30, 40, 50 times over the course of a few hours. Now, this is a process that's very fast in the lab, but it's longer as a process in nature. But we have a challenge for PCR, however. Now, in DNA synthesis, DNA helicase is the enzyme that does the unzipping. It separates those two strands that form the DNA molecule. Only when those strands are separate can we copy each respective template strand, but they have to be isolated from one another in order to do that. But we don't use this enzyme in PCR. Instead, we just simply increase the temperature of the solution that the DNA is in to the point where those hydrogen bonds cannot hold themselves together. The temperatures of around 95 degrees Celsius are enough to break these hydrogen bonds that hold the two strands of DNA together and separate them into distinct strands. However, a problem arises because those are the same temperatures that can denature DNA polymerase, that enzyme that builds the new copy of DNA. We need that molecule in order to make our respective copies, iteration after iteration. But if we use temperature to denature and separate these strands of DNA, we will denature DNA polymerase and it will be left non-functional. That, by the way, is the case for the DNA polymerases of most living things. The vast majority of living things have DNA polymerase that will denature at that high a temperature. So think about this. If you had to find an enzyme of DNA polymerase that would work under extremely hot temperatures, where would you look? What organisms might you go to look for if you wanted to find a very heat-resistant DNA polymerase molecule? Researchers had the same question in the early days of PCR technology. Some organisms could survive in incredibly high temperatures, temperatures that were much hotter than the temperatures that we take things to in a PCR. Those thermophiles and hyperthermophiles, those heat-loving species, are a great group to go and look for to find a heat-tolerant DNA polymerase molecule. Now, these organisms can replicate and copy their DNA even under these extreme conditions. So their DNA polymerase enzymes are functional even at these high temperatures. they don't denature under those conditions. So this is where a species called Thermus aquaticus comes in. This is a thermophilic bacterium that was found in hot springs in Yellowstone. Now, this species regularly lives in environments around 70 degrees Celsius, but the proteins that it has can tolerate much higher temperatures than this without denaturing. So today, modern PCR uses a special enzyme called Taq polymerase, otherwise known as Thermus aquaticus DNA polymerase, the specific DNA polymerase that we got from that species. So let's talk about how a PCR works. I want you to, as we are learning about a PCR, compare it against what you now know about DNA synthesis. So first, we have to unzip the DNA. To do this, we heat our sample of DNA up to around 95 degrees Celsius. And the DNA unzips because those hydrogen bonds that hold the two strands together break. As a result, you end up with two distinct starting strands of DNA. Then, the next step is to let site-specific primers bind to the DNA. So, think about it like this. You have a specific region of DNA that you're interested in. If you can make a little tiny piece of DNA that complements a region directly upstream and directly downstream of that region, you can introduce those little primers into the environment here, and those primers will find their complement when the DNA is unzipped. They will find their match, and they will bind to it, and that will be the starting point for where DNA polymerase begins construction. Finally, it's time to actually build that complementary strand. And so Taq polymerase is going to come in, bind to those DNA primers, and start building the complementary strand. When it finishes, it reaches the end of that sequence, it stops, it detaches, and then we can start the whole process all over again. Rinse, repeat, rinse, repeat. We do this multiple times, each time doubling the number of DNA molecules that we have in this sample. So if we start with 1, we will double that to form 2. Then we will double that the next time to form 4, and then 8, and then 16, and then 32, and so on, and so on, and so on. This is a breakdown of what that process looks like. Okay, so here we have the DNA template at a normal temperature. We increase the temperature to do the step that we call denaturation. We go all the way up to 95 degrees, and that will cause the DNA to split into two distinct strands. Then we bring the temperature down a little bit. We have the primers. The primers anneal. They attach to their respective complementary sites on these different molecules. That is the annealing step. Then we increase temperatures just a little bit to help Taq polymerase function, and we do the extension step. We go up to 70 degrees Celsius, and we allow the Taq polymerase to build the complementary strand starting from those primer regions. Then we increase temperature back up to denaturation. We split these two strands that we just formed into two distinct strands. And then we repeat the process. Denaturation, annealing, extension, denaturation, annealing, extension. Repeat, repeat, repeat. We actually have really cool technology today that can do this whole thing for us. Way back when, this used to be somebody's specific job that if you were doing this research, you would be carefully watching over your samples for almost an entire day continuously and modifying these temperatures yourself. But today we have really cool machines called thermocyclers that can cycle through these different temperatures all on its own and facilitate this process. Now, today, the process takes maybe 4 hours.